So let's begin our review of this 2025 AP Biology FRQ. This work that I'm doing here, my explanations, of course, is not endorsed by the College Board. I have nothing to do with them. I'm a current high school bio and chemistry teacher. It only represents my personal interpretations of the problems. My focus here is to explain the questions because I use these questions as worksheets for my up-and-coming classes. So I'm not just here to tell you what points you may or may not have earned for a particular question. I'm really here to explain the questions as if you're trying to learn from them. So understand that as I might over-explain the questions, okay, and have less focus on possible what points you earned. I'll go over what I think the AP was talking about in this scenario. Remember, in 6 months, they'll release the key or less than that, and you can see how well I have done anyway. So any case, let's be crazy and start in order with number one. So here we go with number one. Most proteins are secreted from the cell, must be transported to the endoplasmic reticulum. Hey, that'd be called a rough ER if it's got ribosomes, right? And of course, protein production or translation requires the rough ER, either during translation or after translation. That's a big point here. Okay, this entire question is about proteins being made either during or after translation. And that's what the focus of this study is. And again, this, as much as it is a biotest, it's a reading test, as we're going to give you this scenario. So in any case, they're going to throw a question at you, and sometimes it's just a, pure informational question, knowledge question like this is: describe the functions of ribosome. Notice they didn't say rough ER, they said endoplasmic reticulum. So they didn't mention at all anything about ribosomes. But some background information you need to know is that the ribosomes are these two subunits that come together and clamp down on a messenger RNA, that single-stranded transcribed code that came from the DNA. And with the help of the two subunits, it allows for the tRNAs with their anticodons to bind to the messenger RNA in particular positions within the ribosome. And the ribosome also acts as a catalyst to move these amino acids from the tRNAs onto a longer chain, and allows for the exit and the bringing in of other ribosomes. So it essentially, describing the function of ribosomes, it helps, it facilitates translation. Okay, and I'll give you my complete answer on the bottom here in a second. And this, pure knowledge question, I said the function of ribosomes is to facilitate the translation of proteins by combining the two subunits of the messenger RNA and providing a framework for tRNA to enter and exit. I probably added way too much here, okay? Just to talk about the fact they allow translation to occur. The idea that I said facilitate would be enough. But again, I'm here to teach. And also, I want you to understand that the big subunit, the larger subunit that goes toward where the tRNA can enter and leave, and those EPA positions, as you learned in translation, that framework allows for the tRNAs to enter and leave, bring amino acids with it, and there's also some catalytic function of that RNA and protein structure, that ribosomal RNA and protein structure, that allows for that translation to occur, to go from a code of RNA to actually amino acid chain. So again, I'm giving you more information as a background, but really the idea that ribosomes facilitate the translation, or they help with providing a physical mechanism for which proteins can be made from RNA, is essentially enough for that question. So let's go on to question for 1B. So 1BI here says identify the dependent variable in the experiment shown in figure 1. Figure 1 is right above, so easy to look at. Looking at the title may give you some hints. The average relative amounts of the Sec62 and SR proteins in cells treated with control types of SI, okay, small interfering RNAs that are regulatory RNAs, okay, that affect, of course, eukaryotes, that kind of control there. And so if you read it, you see that there's some value that we're measuring, which is the amount, and there's a treatment group treated with these types of regulatory RNAs, the siRNAs. So when you think about it, who affects who here? We're looking for the dependent variable. So what's going to depend on what? Well, the amount of protein produced depends upon, how much of the regulation that you have going on. The regulation, of course, is due to the siRNA. So the amount depends on that. The amount of siRNA that you're adding to treatment groups does not depend upon the protein amount. So that if you say that to yourself back and forth, you can find out who depends on who. And by the way, when you look at a graph or any type of presentation of the data, the y-axis, okay, is going to show you what we're measuring. We're actually measuring the amount of the relative protein content of these two different types of protein under different what? Different treatment groups. So what we measure is the dependent variable. What's on the y-axis is the dependent variable. Okay, the treatment groups, these are the independent variables. These are the things that we manipulate, have less of, different types. That's independent of what's happening. All right, so we're going to say that the amount of protein depends upon what kind of treatment group you put it under, as you can see here. So in 1b, the relative protein content of the Sec62 protein and the SR protein are the dependent variables. Simple as that. Moving on to 1BII. BII says justify. That's our action word. So we're going to have to give some support for something. Let's see what that something is. Why the researchers included the control of measuring the relative amounts of both the Sec62 and the SR proteins in cells that were treated with the Sec62 siRNA. So they're pointing us right here. And this is exactly what they're pointing us, because they're saying we have Sec62 and the SR proteins treated with this specific regulatory RNA. And that confused a lot of people, because people say, wait a minute, I see the control here. How can this be? How can this treatment group be the control? Well, actually, all of these are a type of control, because you need to understand, although this is a biotest, it's a reading test, and the setup here is very important. So the blurb above the data is talking about the idea that researchers are trying to figure out, okay, which type of transport protein is responsible for the different types of translation that occur. We know that proteins are essentially very polar. So to get into the endoplasmic reticulum, they're going to need some sort of transport protein to allow that to happen. Now, very important, they said already in the reading that there's two types. There's translation that completely occurs outside of the ER, and then it gets moved in. And then there's translation that occurs partially, and then it gets completed in the ER. And the researchers are trying to figure out which proteins are responsible, okay, for those two types of processes, and whether or not two different proteins do those different jobs. So that's what this is about. All right. So these proteins that we have, Sec62 and SR, are nothing more of, these are the transport proteins that are helping the type of either partially translated proteins or completely translated proteins enter through that channel, so to speak, and get into the ER. Okay, which they finally have to make a vesicle and go to the Golgi for complete finishing or folding and processing. So it's important you understand what's going on here. So this is not the entire, okay, experiment. It's really to prove that the regulatory aspects of this experiment are in fact working. Okay, that's important. And so when you understand that, why in the world why did they do this treatment group? Well, think about it. Look at the outcome. When we applied the Sec62 siRNA, it significantly decreased the Sec62 transporter protein. There was less of that protein being made in those cells. And when I grew those cells along with, and I put that RNA, that Sec62 siRNA into the SR proteins, it had no effect, possibly maybe even increased it. So what can we clearly see? There's a significant, look at the error bars, there's a significant, okay, change here. Now understand, in this question, the error bars are standard error of the mean plus or minus 1. So even when I consider plus or minus 2, okay, there's still going to be, I double these, no overlapping. There's a significant decrease in the production of the Sec62. What does that mean? Why would we do this? We're going to prove that the Sec62 siRNA inhibits the Sec62 protein for the next test. Now they picked out this one, but they could have picked the same question. Why did they give the siRNA to this group? And notice the siRNA negatively impact significantly, well, maybe not if you consider double, but you can see that it may, if we considered plus or minus 1 error bars here, that there's a significant or a possible significant decrease in the SR protein. That means that basically we're proving that the siRNAs in fact inhibit the expression, okay, of the proteins that are the transporter proteins that we're going to study in the next piece. So they pointed us here, but they're calling it a control with no number, but really what it is a positive control. We expect that the regulatory siRNA, the reason why it works is because it binds to the RNA for the Sec62 protein and prevents translation. So we would expect it to decrease, and it did that. That's a positive control. We're getting what we expect. Okay, so we're proving that the regulatory aspects of this experiment do in fact work. And even though they're saying control here, they're just saying that we're adding just any old siRNA that's not specific for each one of these proteins. And that's also proving that the siRNA has to be what? Specific to actually decrease expression. So all of these are controls. In fact, all 3 of these tests are positive controls. You didn't have to talk about that, but let's get to my answer. So you really had to get into the reading to understand what was going on for this. Let's get to my answer. So 1B, I said that the researchers included the control of measuring the relative amounts of both Sec62 and SR proteins that were treated with Sec62 siRNA only because it was necessary to prove that the Sec62 siRNA will selectively reduce the expression of the Sec62 protein only. This positive control allows the researchers to determine which protein is transported during and after translation in the second study. We needed to know in the second study which transport protein is actually doing the job on these different proteins. And to eliminate one over the other would be helpful in making that determination. Therefore, we needed to know that certain siRNAs will in fact eliminate or lower that expression. Very important positive control, that first study in figure 1 for both. All right. So again, I know people got lost in the sauce there for a little bit because I said control and people wanted to look at the first treatment, but they're all positive controls and you needed to read and understand the process there. So that to me was a little bit tricky. Okay, this is not going to be an answer, in my opinion, where you can say in other questions and other FRQs when they say, oh, why is there a control? Oh, it's easy for comparison, okay, or to eliminate other variables. That's not what this is about. This is talking about reasoning for a positive control, in my opinion. But we'll see what's produced here. So I thought this was a little bit tricky, but you needed to understand the process. On to 1BII. BII says, based on figure 1, make sure you go to figure 1. Here's our describe. Describe is like giving characteristics. The effect of the production of SR protein when cells are treated, okay, SR protein, when cells are treated with the Sec62 siRNA. So we're still looking like BII, if we were just from this question, we're looking at this treatment group. Okay, so what is the effect of the SR protein? Okay, which is this one right here, when cells are treated with the regulatory protein, a regulatory RNA for the other protein? Well, there's a significant difference or increase in production as compared to the Sec62. And this, again, okay, very careful with this question, all right, because the significance, in my opinion, depends upon plus or minus 2 SEM. Now, if they come out in a couple months and say that, okay, no, we didn't mean to say that, okay, but there is definitely, when you double the error bars, because this was a plus or minus 1 standard error of the mean, we usually use plus or minus 2 because that gives us the 95th percentile. But even if you double the error bars here to do plus or minus 2 SEM of the mean, now you can still see there's a significant difference. Okay, so when we describe the effect of the production of the SR, it's significantly greater. Okay, and remember, careful of the wording. Their wording is horrible for me sometimes. Describe the effect on the production. Well, it seemed to be that the Sec62 siRNA had little negative effect on the production, may have contributed to some positive effect because there was a significant difference or increase compared to the Sec62. I'm comparing it to here, and I'll also can say I can compare it to the controls, the other control when you're using control siRNA. So let's go see what I said. So I said the production of SR protein when cells treated with the Sec62 siRNA is significantly higher than the Sec62 protein production in the same treatment group. I also added it's also significantly higher when you consider plus or minus 2 SCM than the SR protein produced in cells treated with the control siRNA. Were they after that? They said describe. So they may just take significantly higher. I added a lot more. They probably would take an increase, that the effect, remember, it described the effect of the production given that treatment. Maybe you can say there's a increase. Okay, I said significantly higher, or a significant increase. Again, the wording, be careful with. They describe the effect of the production. So I said the production when treated is significantly higher, or it's significantly increased, or of the other. And I always love to add, when I'm comparing, I want to talk about both. Okay, it's higher than what? By saying it's higher, okay, is not important. Because there are error bars, I'm always going to add significant if the error bars overlap. They overlap at plus or minus 2 for sure. Okay, and then just for good word, I'm going to compare it to the other treatment group that had the non-regulation, or just a, how can I say it, a random siRNA that wasn't specific. So on to 1C. So in 1C, we're talking about identifying the independent variable in the second piece to this study. This is actually the study. For those that are joining me for this question only, this question had another piece to it, okay, and that piece, of course, is this graph, which is basically a control graph, a positive control graph that we talked about, to show that the small interfering RNA, okay, is specific to the individual types of transporter proteins that we're testing. Okay, when you had a non-specific siRNA, there was no significant difference, but when you had specific siRNA, there was a change. We can argue the significance of that change based on plus or minus 2 SEMs here, or plus or minus 1 SEM, but you can see that there's a difference. There's clearly a repression of that protein by these RNAs that are specific. So those are positive controls. Okay, so that's not really the study. That's just proving that the components of our study do what they're supposed to. Okay, and that's why it's a positive control. Okay, here we are at CI. Identify the independent variable in the researcher's second experiment. All right, Now, to me, this is the real experiment. The first experiment was just to identify the siRNAs that actually manipulated or stopped or controlled or regulated the individual transporter proteins. But in any case, here we go, identifying an independent variable. Now, the independent variable is the variable that's in the treatment groups. It's the variable that we are going to manipulate, and then we're going to measure to see how well something does from that manipulation. Remember, on the y-axis, this is what we're measuring, and that's the dependent variable. Those amounts depend, okay, are upon what kind of treatments you put them under. So if you look at this question, it says the figure 2 says average relative amounts of 3 proteins that were transported to the ER when treated with the control siRNA or the Sec62 siRNA or the SR siRNA. So essentially, the independent variables are nothing more than the siRNAs that are being used to treat the 3 different proteins. And we're doing that right here. So let's go take a look at my answer as written. So I stated the independent variable is the Sec62 siRNA, the SR siRNA, and the control siRNA. Those treatment groups. Okay, moving on to 1 CII. CII, here we go. Based on figure 2, that's the one right in front of us here, identify the proteins, and I love the identify action word. We're just going to have to do what? Go find it and tell me something. They want the protein or the proteins that when treated with the Sec62 siRNA showed an increase in percent transport to the ER compared with the control. So I see error bars here, and I'm looking for the standard error of the mean. Oh, it's plus or minus 1 standard error of the mean. It's not talking about plus or minus 2. We should recognize the fact that plus or minus 2 standard error of the mean in error bars is 2 standard deviations of where the true mean could possibly be based upon your sample size. And plus or minus 2 standard deviations around the true mean is a 95th percentile. We like 95%. So normally something is significant if we're outside that 95th percentile. We call it abnormal or outside the norm. So normally it's plus or minus 2. So this is an interesting question. If I don't consider using plus or minus 2 and just use the error bars given to me, then based upon the data given, it looks like protein 2 is significantly higher than the control when you add Sec62. And then in protein 3, when you treat it with the Sec62 siRNA, it's also significantly higher. So to me, that'd be protein 2 and 3, no if, and, or buts, if this was plus or minus 2. But are they trying to be tricky here? This is very interesting. So given this information, and given the information that we should be outside the 95th percentile, I have to answer this by saying there is no protein that is significantly or has an increase, because when I do plus or minus 2, okay, that means this error bar is going to go double plus or minus. And you can see that even if I double this one, we're going to overlap. Same thing over here. I'm going to double this one. I double this one as well, and we're going to overlap now. So the overlapping of the error bars means that there really is no significance when you consider the plus or minus 2. I don't consider what's happening in protein 1 because this is a decrease. Now, when you think about it, if I, again, double the plus or minus, there's no significant what? Decrease there. So there is no protein, according to the statistics that we're supposed to understand for this course, that is higher when you give the Sec62 regulatory mechanism. Okay, so If we do consider that plus or minus SC, and again, it's going to be interesting what they accept here, if that's going to be something that is going to be used to verify, or they made a mistake and they'll accept either way, again, 2 and 3 would be the answer. So interesting what they're going to do here. To me, the answer, based upon how this is presented, is that there is no significant increase for any protein treated with the Sec62 siRNA. Okay, so that's my answer, and I'll show you what I wrote. So I said in 1CII, protein 2 and 3 show no significant decrease to the ER compared with the control when treated with the Sec62 siRNA. And of course, I made this little statement here. When you consider the SCX plus or minus 2 values, okay, if you consider that's our standard. And again, not sure how they're going to treat this. I would think that that's what they're after. Okay, in any case, onto 1CII, which is a nice little calculation. So CII, protein 1 is encoded by 234 nucleotides, and protein 2 is encoded by 495 nucleotides. Assuming all the nucleotides for both proteins encode amino acids, that means we don't have any stop or start codons, right? Calculate the difference in the number of amino acids between the two proteins. So very simply, we know that a triplet codon of 3 nucleotides makes 1 amino acid. So we're essentially dividing by 3 and then subtracting the difference. Let's go see the calculation. So I got for 1CII protein 2, the 495, I divided by 3 because of the idea that there is a triplet codon. There are 3 nucleotides that code for each amino acid. And I got a total of 165 amino acids that were which that code would represent. And for the 234 divided by 3, I got 78. And they wanted the difference, so I subtracted and got 87. Okay, that's pretty straightforward. Onto, I believe, D. DI. Researchers claim that protein 1 is the only tested protein that's transported to the ER following its complete translation. Hey, that's in the reading. And in the reading, they talk about that there is a type of transporter protein that will help move the completely translated protein from the cytosol into the ER. And that's going to be the Sec62 transporter protein that's responsible. They told us that. So it should make sense to you if we can somehow negatively express it or decrease the expression of Sec62. And of course, the way that we're doing that is with a small interfering RNA that's specific for Sec62 called the siRNA 662. If we would treat that with that protein, with that siRNA, we're going to decrease the Sec62, and if there is a protein that completely translates and needs the Sec62 to get into the ER, it will have a hard time getting into the ER because there just is not enough of these proteins to help them move in. So what we're looking for is a decrease in the amount of Sec62 in protein 1, and that's what we have here. In fact, we actually have a significant decrease. Okay, so there is the treatment group using the siRNA for the Sec62, and look at it. It is negatively affecting the protein. Remember, don't forget what you're doing here. We're measuring how well the protein, or how much of it, is found in the ER compared to the control. So there's a significantly less amount. If you look at protein 2 and 3, whether you want to argue standard error of the mean plus or minus 2, this is the only protein that has a significantly less amount of the protein 1 in the treatment group where we treated it with Sec62. When we treated it with the SR, there was probably almost no difference with the control, but it was definitely decreased, okay, where we essentially lowered the expression of the transporter protein, and therefore negatively impacted the amount of protein 1 that could go there. So what this is telling us, this treatment group with my arrows, this treatment group right here is screaming because it is being, the protein 1 in this treatment group is being negatively impacted. Means it has a tough time getting into the ER. So there's a lower amount. And why? Because we lowered the amount of Sec62 by using the small interfering regulatory messenger RNA that inhibited that translation of that protein. It helps it move in. So again, kind of a little bit confusing because we have different things going on. We have a transporter protein. And if you're getting lost in the sauce, that this is like let's talk about, like, this is a transport protein right here. This is my passageway here, okay? And this would be the ER. Let's do it this way. And we have this perfectly translated protein, and it only can get through this type of protein, which is, in this case, Sec62. Let's write the 62 there if you can. I can write it here better. So this is the 62. All right. And so if we lower the amount of that, then this protein cannot get through the ER, and there's less of it. So this is protein number 1. Don't forget, when you compare that to the control, which basically had a small interfering RNA that was just non-specific, it was totally unaffected. And when you use the SR, now that's the other protein, the other transporter, that's for when it was partially translated and then had to travel in, there was very little difference. So the idea, very simply, by negatively impacting the amount of protein 1 found in the endoplasmic reticulum, is because it couldn't travel through, because there wasn't enough of the Sec62 protein expressed. So that proves that protein 1, all right, was completely translated and couldn't get in the ER without the Sec62. All right, so let's go to my answer, which is probably a little more comprehensive and clearer than my kind of explanation here to make you understand this. So I said in 1D, because Sec62 protein is needed to transport the protein after translation, and the treatment of the Sec62 significantly decreases the relative amount of protein 1, while proteins 2 and 3 had either no significant or were significantly increasing. That's depending upon if you take into consideration the plus or minus 1 or plus or minus 2 standard error of the mean. The researchers' claim is definitely supported. Okay, so the 2 and 3 didn't have a significant decrease, and I can say significant because the error bars, okay, even with plus or minus 1, would support a significant decrease, right? If I double these error bars, then even by doubling them plus or minus 2, it still would be no overlapping there. So on to the last question of this first overall question 1. Question 1DII. For any protein it enters the ER, researchers claim that amino acids close to the protein amino terminus, that just means the ending part of the primary chain that was just made in translation, okay, determine how the protein is to pass through the protein channel. They did talk about the fact that for these proteins to enter the ER, they have to go through a protein channel. That was in the reading, okay, within the ER membrane. So this whole question's about either partially translated or totally translated proteins entering the ER, and obviously through a protein channel. And there's different types of proteins that we tend to remember, the SR and so forth, but that's not what they were getting at here. They want you to justify the claim, and this is a really good part here, based on your understanding, okay, of factors that affect the transporter proteins across membranes. So what we do know is that in our cell membrane, or even in an ER membrane, remember these are membrane-bound organelles, so they have the same type of membrane, where they have a hydrophobic interior, and we have this phospholipid head here that's hydrophilic. What we know that cannot pass through the ER membrane is large non-polar macromolecules like proteins. Proteins, even though we will designate the type of amino acid by their R groups, right? All of these amino acids have a particular type of R group that we label as hydrophilic, hydrophobic, or ionic mostly. We know that amino acids in general are just too big and too polar to pass through. So that we're going to need some facilitated approach or facilitated diffusion here through a protein channel. And so when I look at this question, your understanding of proteins is that, well, we know that proteins are too large and too hydrophilic to go through the channel, diffuse through the membrane itself, so they have to go through a channel. Well, that channel has to be mostly hydrophilic because amino acids in general are all hydrophilic. And for those that are not understanding me, if you look at the general shape of an amino acid, you've got a carbon double bonded to an O, which is a very polar shape in itself. We have, let's just say we have an O here, we have an alpha carbon. Now this arc could have hydrocarbons or be nonpolar itself, but the rest of this molecule with the nitrogen and one end, and it could be bonding to a peptide bond. If I'm just drawing a very simple amino acid, we can see these two O's, whether they're the terminus end or bonded in a peptide bond, are going to be a negative end, be a polar part of the molecule, and this end here also. So we know that amino acids generally are very polar. The R groups themselves talk about how they interact with themselves. So in any case, if you got this wrong, it's because very simply we need to understand that proteins are polar, and therefore they're hydrophilic, and therefore they have to interact with that protein channel. Okay, and don't forget that protein channel is going to span the entire membrane of the ER. And we're talking about is in this interior area here, okay, or I'll shade it or color it a different color. This interior area here has to be mostly hydrophilic for that protein to travel through that. Okay, that's the whole point, because it can't travel through the hydrophobic, okay, interior of the ER's membrane. All right, so that would mean that I would think for this to totally pass through the ending, okay, or they say the protein's amino terminus, if that got you in the wording, just means the terminating end of the amino acid chain, should also have R groups, okay, that are mostly hydrophilic. If they're hydrophobic, they may not interact with this interior of the channel, and it might get stuck. So the wording to me would be, or the answer here is understanding that the protein channel has to be hydrophilic, and that the R groups coming off these amino acids should also be predominantly hydrophilic, especially at the end, so they don't get hung up and stop. All right, so let's take a look at what I exactly wrote there. So I said 1-DII amino acids that have hydrophilic R groups near the polypeptide terminus will allow it to move and interact with the hydrophilic protein channel, while hydrophobic R groups would not. Since the membrane has a, I should say the ER membrane has a hydrophobic lipid interior, the protein channel needs to have amino acids that have hydrophilic R groups in order for large polar peptides to pass. So the bottom line is that these large macropolar molecules cannot pass through the ER that's mostly hydrophobic. Just cannot do so. So we need a protein channel, and that protein channel has to be hydrophilic, because like dissolves like, it's going to interact. All right, so therefore we would expect that the ending of the primary chain of the polypeptide that was just translated should also have hydrophilic R groups to prevent the repelling of hydrophobic behavior with the hydrophilic interior, so that this polypeptide can pass. All right, on to number 2. So number 2A. Many insects rely on pheromones, which are chemical signals that are released by the females to find mating partners. Scientists hypothesize that in a certain type of moth, the behavior of male moths in response to pheromones is regulated by an extracellular signaling molecule 20E. So this little part tells me that this question is about a transduction signaling pathway for a molecule they're going to name 20E that essentially allows for the pheromones to produce some sort of cell response that leads to the moths, okay, apparently moving toward females or mating with females or finding them, okay, since a pheromone is a chemical signal that's going to travel across an ecosystem, apparently. Okay, so the question underneath this little statement says many receptors are embedded in the plasma membrane. So I kind of highlighted that because it's important to understand that this is a molecule that's not going to get 20E into the cell. It's got to hit a receptor. And essentially what they're saying is describe. I like that action word, describe. So tell me something about what? Oh, the polarity. Tell me something about the polarity of the portion of the receptor that's inside the membrane. Well, we should know that in our plasma or cell membrane, I'm just going to draw down here where we have these phospholipid heads that are polar, and we have these non-polar lipid sections that could be saturated or unsaturated. In order for to have a receptor, normally we have some sort of protein that has some glycoprotein or some sort of receptor. Of course, I'm making this oversimplified. And that receptor has to be attached to a protein. So we're assuming we're talking about the rest of the protein. And for it to be able to work in the interior, or at least span the length of a cell membrane that has a huge hydrophobic area in the middle here because of those lipids, those lipid tails, we should expect that for this to sit in here, that the attractive forces in the interior should also be hydrophobic, as like dissolves like. So it's as simple as that. So I would say that the polarity of the portion of the receptor is going to be nonpolar so that it can interact and stay in that area. Okay, and often the area here would be polar, so it kind of fixes itself in that area. Okay, but they're talking about the portion of the receptor that's inside the membrane. Inside the membrane is hydrophobic. And so it is one word. Okay, and so therefore for that protein to be able to stay there and interact and kind of lock in there, it has to have similar attractive forces that hydrophobic would have for itself. Like dissolves like here, hydrophobic, hydrophobic. So we would say that the portion of the receptor is nonpolar. Let's go see what I wrote. So I said essentially that the portion of the receptor that's inside the cell membrane is nonpolar. Didn't ask for why, they said describe the polarity. So you really could have said it's nonpolar, and that would be the end of it. On to 2B. 2BI. Using the template in the space provided for your response, construct an appropriate type of graph that represents the data in Table 1. Your graph should be appropriately plotted and labeled. All right, so we need a graph, okay, based upon this data table here, data table 1. And when I look at this table, I can clearly see that we're measuring either the general activity or the orientated activity. So what I underlined here is the dependent variable. The dependent variable, of course, is something that we're measuring. And you may say in this question, hey, they didn't ask for a dependent variable. If I'm looking to build a graph, I've got to know what's being measured. What's changing in response to some test group or stimulus? And what is changing in response possibly to something? That's what we're trying to figure out, if there is a response to a new stimulus or a new test group or a new environmental change. That's my dependent variable. We're measuring how much general activity or orientated activity is happening in the moths. General activity, of course, is just a movement of the moths that is, I guess, quoted general, not related to any particular direction, whereas oriented activity is movement in a certain direction perceived, I believe, toward the pheromone. So understanding that, when I build a graph, now I know that what I'm measuring is going to be on this y-axis right here. So the y-axis for me is the dependent variable. So it's the activity. Now, in this particular set of data, if you look very carefully, it says percent of total time observed. All right. And here it says percent of general activity. Okay. So you can see that these dependent variables are not quite the same. All right, so I do believe that is a particular piece of information you have to take in consideration. So what you could do is you could write two y-axes here. You could write the general activity on this y-axis, and you could write the orientated activity on that y-axis. Now, interesting enough, they share the same kind of span, or I would say range. So if I'm going to build a graph I'm going up to 95 plus or minus 5, in this case plus or minus 2 standard deviations, and this is going from 60 to 25. So I could build a graph up to 100 and use the same data lines, meaning I could go 10%, 20%, 30%, and I could use the same values over here. Sometimes when you have to write another y-axis, okay, you have to write a new grid or a new spacing of numbers, which is the same thing, to interpret where the data points go there. But in this case, we're going to share the same grid lines for the same percentages. And that's why I also believe that you could have also shared the same y-axis. You could have said, hey, this is representing of general activity, and you could say it's also orientated activity, all right, with the percent of general activity. It's not really that important in my eyes. I do believe that you could have shared the same y-axis. Now, if you just wanted to be very careful, you could have put them in both. I do believe writing them both ways would be fine. And they don't go in any particular lengths for you to understand its percent of general activity, in my estimation. So I do believe you could share the same axes, the same y-axes, and as long as, in my opinion, you do put both general activity and oriented activity, okay, on that y-axis. But we'll see when it comes out, see what they accept, all right? And of course, you must also, okay, for those that are still getting this wrong, you must include your error bars. In this case, in this set of data, they did give us plus or minus 2 standard error of the mean, plus or minus 2 standard error of the mean. Okay, so let's take a look at my graph, which you probably saw in a previous question. Again, your graph may look a little different based upon the actual grid of lines they gave you. I created my own set of lines, okay, based upon what I think could have been given to you, but I'm sure it's very similar. And so if you notice, I did 2 y-axes, and I described both of them. And I again do believe that you could have put both on the same y-axis as long as you described both of them there. All right. Very important, in my opinion, that they need to be described. You just couldn't put activity. I don't believe that writing activity here, you'd get the point for the labeling. All right. So again, because they shared the same scale, remember, 0 to 10, 110 I have here, and 0 to 110 here, I do believe that you could have used the same y-axis as long as the label had both labels in them. All right. And of course, you needed to have your error bars. I've noticed in previous tests, there's a point for making sure your error bars are labeled and of course plotted correctly, and of course labeled. This is normally 3 points, making sure that you plot this correctly, that you have a appropriate scaling and labeling. So somewhere in there, there's usually 3 points. Now, I always put a title in, just as something that I've just been doing forever in science. should always have a title or some information that describes what your data table is. That's not being looked at for a point, but I just kind of have it for me. So that's the data points there. All right, and that's the graph. And I think I've covered, what they're looking for here. And again, what they're exactly going to grade here and how they handle that double y-axis or just sharing, I do believe that could be both sides there. Okay, on to 2BII. 2BII here, based on the data in Table 1, determine the type of activity. It's really important to reread and rephrase, if you have to, these questions, because these questions are particular. They're telling you exactly how they want you to answer them. So again, determine is our action word here. All right, so we're going to find out somehow through the graph, determine the type of activity that was affected by inhibiting the expression of the dope ECR receptor. Of course, that's being inhibited by the small interfering ribonucleic acid that's specific for that receptor that accepts that chemical message or messenger that is part of the dopamine pathway, right? So that's important you understand that dopamine transduction signaling pathway, I should really suggest. So the best way to do this is go look at the data. So notice they set you up in B1, make a graph with the data, and now they want you to look at that data. So it's easier to look at it. If you didn't build the graph for whatever reason, you could look at the plus or minuses from the standard error of the mean, which is really what you need to look at here to determine if something was affected. But the way that they ask is determine the type of activity. Okay, well, there's two types. There's orientated and there's the general activity. Which one was affected? They didn't ask affected how. So it's really hard to, really what you want here is that, well, one was affected, one wasn't. So to me, they could have said identify, but that word determine means that you're going to figure it out through the graph. Okay, so interesting why they're saying determine and not identify here. Okay, that what type of activity was affected. Well, one was affected and one wasn't. Let's take a look at that in the graph. So if we look at the general activity, where we had the saline, the salt solution, right, that was added to cells, and then we had the siRNA, the regulatory mechanism here to try to eliminate the expression of that gene. We can see because of overlapping error bars, there is no effect. All right, it's the general activity was not affected by giving the group of cells, the moth cells, the siRNA in terms of their general activity. But because of what? The error bars not overlapping here, unlike the one we just looked at, we can see that there is a significant decrease, okay, in the change of the orientated activity due to the siRNA, which is lowering the expression. So I said here in 2BII, the answer, the orientated activity males was significantly decreased. Now, I think I'm giving more information than I need to. They said determine which activity was affected. When you think about it, well, it was the orientated activity that was affected. So interesting how they word that. So just to cover all bases, I'm going to say the oriented activity of males was significantly decreased, and I underline that because if you're basing it upon error bars, you should talk about significance in my book. So I really believe you need to say that it was significantly decreased. Now they just said which one was affected, but I'm going to say not only was it affected, I'm going to determine that it was decreased. I feel like determine is more than just identify. So I'm going to say significantly decreased as compared to the what? Saline control. If it's decreased, that's decreased from what? How is it affected? Now that they just said determine what was affected, what activity. Now we're going to say that the orientated activity was affected because the error bars don't overlap compared to the control. And I'm just going to go ahead and say it was significantly decreased, just to give that little extra information. And I'm also going to compare it just for good, practice to the saline solution to control. To me, you should always talk about the significance of a result compared to some control. So I'm going to compare it to the saline control here, when siRNA inhibited the expression of the dope ERC receptor. So again, clearly there is something being affected. It was the orientated activity that was decreased or affected. Again, I feel like you can just say it was affected, but again, when you say determine, I'm thinking you need to be a little more deeper here. So just to cover all bases, as again, I'm going to say significantly decreased, and I'm also going to compare it to a control for good measure. All right, so that's clearly what changed here. And to understand what's going on, clearly the orientated activity of moths, who of course are now going to be moving toward the pheromone. That's basically what the orientated activity is that kind of motion is being decreased in the presence of pheromones because they don't have the receptor to accept that chemical message to elicit a cell and organism response to move in that direction. All right, so on to the next question here. Number 2CI, based on table 1 or the graph that you built, identify the treatment group which the oriented activity was greater than 50% of the general activity. So all we got to do is see which of the two treatments, because in the orientated activity treatment groups, we either have saline solution or the siRNA treatment solution, which one of them is greater than 50%? Really, it's that simple. Now, you may say, what about this general activity business? Well, remember that the oriented activity was based upon that general activity. So if we scroll back up, okay, to this information, which I keep scrolling away from, here we go. We can see that the orientated activity is a percent of the general activity. So you can just read, really don't need the graph. Okay, you can see that the treatment group in the oriented activity that's above 50% of the general activity, this is what's measured. It's measured based on the percent of general activity. That's really important. Okay, so we're looking for something that's greater than 50. And it would be, of course, the treatment group with the injected saline solution. All right. That's the treatment group here that we're talking about. Again, you want to look at that in terms of the graph, we can do that. So if we're looking at the graph, we just have to identify again which treatment group. There is only 2 treatment groups for the orientated activity. And remember, we measure that by percent of general activity. So that's actually what it was measured against. And you can clearly see that the saline solution, okay, or the saline treated group of moths, is 60%. Even if you consider the plus or minus of the plus or minus 2 of the standard error of the mean, okay, it still is still over 50%. You need to look at if you look at the minus position here, so it's still above 50%. So we would say the saline solution, and they want just to identify. We love. Who doesn't love the identify action verb? Just give me. So all you need to say is the saline oriented test group. That's it. All right. And I just continued and said it was more than 50%. Okay, but that's all you needed to do there. So hopefully that makes sense. And that really should be a gimme. So if you missed that, you got to clean that up. Okay, just reading right off the graph. So on to 2CII. So 2CII, the scientists studied some moths with a mutation in a gene encoding the gene protein. The mutation prevents, this is important, really important, the mutation prevents the GTP from displacing the GDP. That's important there. Bound to the G protein. So somehow we're preventing the GTP from displacing the GTP in this signaling pathway mechanism Based on figure 1, we're in a different location now. We're not talking about the data now. We're talking about figure 1. So now we're talking about this signaling pathway. They have a diagram that we're going to go to. They want you to predict. We like the predict action verb. Usually predict is stay the same, go up, go down. We're not explaining. Okay, the effect of this mutation on the what? On the orientated activity in male moths exposed to the pheromone. Well, we know the pheromone, okay, elicits some kind of chemical messenger that attaches to a receptor. That receptor undergoes a transduction signaling pathway to the interior of the cell, right? That signal is transducted into the interior of the cell, and then it leads to that cell response, which is the orientated activity. The question is, what does preventing GTP to GTP in the middle of this process do? Okay, so let's take a look at our diagram here, figure 1. They're telling us to look, so we should look. Okay, so we go there, and we can see we have reception of the 20E molecule here. That is the chemical symbol we're assuming from the pheromone, and that binds, and this reception will lead to an active G protein that will lead to increased expression of genes associated with the orientated activity. But how does it go from the extracellular side to the intracellular side? Well, somehow a GDP has to be displaced by a GTP, and what they're telling us is this is not going to happen. The mutation prevents the displacement or the GTP binding, okay, prevents this process of GTP displacing GTP. It prevents the GTP from displacing the GTP. And if this is the case, we are not going to have an active G protein. And that means we're not going to have increased expression of genes, and therefore there's going to be a decreased orientated activity because of that lack of expression. It is just that simple. Let's get to the written answer. So I wrote for the 2CII, which again is predict, there would be a decreased orientated activity in male moths exposed to the pheromone if there was a mutation in the gene encoding protein. So again, I don't need all the rest of this. All you need to tell me is that there is a decreased orientated activity. That's the prediction. It's going to go down. Okay, there's going to be less expression because we're essentially preventing, even with reception of the signaling molecule in the receptor, we're preventing the transduction pathway signal to get into the intracellular pathway if the GDP does not, or the GTP does not displace the GDP. We can't, there's no process because of the mutation to actually activate the G protein, which is necessary for the rest of the cell's response to continue or be produced through that pathway. Okay, so on to 2D. 2DI. Use evidence from the information provided to support the scientist's claim. So we're going to support, that's our action word. We're going to have to give some information that explains why the scientist's claim is correct. We got to support it somehow. Now the question to me is, what information are they providing, are they talking about? Using the information provided, are we talking about the blurb here? Or are we talking about all the other information that precedes these statements? That's a really good question. So when I look at this, let's look at the blurb here. It says expression of the gene encoding the DOP ECR, that's of course the receptor for the pheromone chemical signal that starts the signaling transduction pathway, is low in male moths during their few days as adults when they're sexually immature. Gene expression rapidly increases as the moths reach sexual maturity. So that's the information. I don't think just from those two statements I could support the fact that gene expression increases the likelihood of males finding females. So for me, I need the information from the study, from the investigation to help this. To me, that first couple of statements there's really not much there, in my opinion, that can help there. So what I'm going to do is go back toward the study and go toward the study here, past the transduction signaling pathway mechanism, and I'm going to find that clearly, okay, that there is a significant decrease from 60 plus or minus 4 to 25 plus or minus 6 in oriented activity. That's the activity that the male moths move toward in response to the pheromone. Okay, there's a lot less of that activity when there is less expression, because the siRNAs will lower the expression, will prevent the translation of those signaling proteins. Therefore, there's going to be less oriented ability or activity and less movement toward the pheromone, and therefore less ability to find the female. So to me, the evidence here is that because when we do negatively regulate and the expression of the gene, there is less activity. So we can expect that if we express it more, don't have this negative regulation, we'd expect to have increased amount of activity, because you're going to make more of the receptor protein that's going to be able to deal with the signal and create that response toward the female's pheromone at a greater amount. So for me, I've got to go back to this, and to the key part is that when it was negatively affected by the siRNAs, the expression stopped. We made less of those receptors, and therefore there was less of a cell or organism response moving toward the pheromone. Okay, so very important for me. So that's, to me, where I need to look at. And then I think I need to talk about the fact that if you're going to express the protein less in less sexually mature moths, then they're going to have less receptors. And then when they become sexually mature, they will express it more and have more receptors. That leads to more of a transduction of the signal and, of course, more activity. That's how I see this problem. So let me try to put that to words. So I said, when the expression of the dope ERC gene was inhibited in the experiment, there was significantly less male-oriented behavior toward the pheromone due to less receptors, right? If it's expressed less, there's going to be less of those receptors to receive that signal. Okay, so that's kind of where I'm leading to here, leaning toward anyway. Okay, and then the last statement I say here, which is probably the most important, thus enhanced expression, and I said, you can see it here, when sexually mature, will lead to more receptor proteins, I should say, or more of the dope ECR protein that will be able to transduct the pheromone chemical messenger 20E, which will lead to increased orientated activity. This increase will lead to more males finding females as they move toward the pheromone that the females release. I probably should add that there. Okay, it's a work in progress as I look at my own work. Okay, so that to me is the best support that I can give, and because they said it's based upon information, I'm assuming it's the information above those two paragraphs. Being sexually immature and not expressing the gene and being sexually, that little piece there does not provide enough, although I did kind of mention that as we go through this. So that to me is the best answer. All right, on to 2DII. DII, based on figure 1, explain how the inhibitor of the dope ECR pathway might serve as an effective chemical to protect crops from moth damage. That's key there, protect crops from moth damage. And he's talked about an inhibitor of this transduction signaling pathway. So figure 1, of course, all right, is nothing more than the figure that describes how this signal gets transduced through the chemical. So if we have some kind of inhibitor, like for instance, we have this work here from a previous question where the GDP could not or the GTP could not replace. So if we have some inhibitor in this process, whether it's at this level or competes for the same binding site, that become competitive inhibitor, okay, or any level in between, if we have something that's going to basically stop this process, then what we're going to have is we're going to have less expression of genes that lead to the transcription of some kind of genes that, of course, perpetuate the movement toward the females as the cell, or I'd say organism, responds. So that's important to understand. So how would all of that less movement toward females help in preventing crop damage? Well, if moths eat the crops and there are less moths, and of course moths are created through sexual reproduction, so if there's less males moving to females because there's less orientated activity due to this signaling pathway being blocked or inactivated somehow, then there's going to be less reproduction. There's going to be less of the second and third and final, there's going to be less offspring to eat the crops. It's just that simple. So let's put that down. So I said an inhibitor of this pathway will result in less 20E signaling molecules binding to the receptor and activating the G protein, resulting in a lower expression of genes that can cause oriented activity in male moths toward females. Now I said less binding, but they still could bind, so I don't have to talk about just less binding. It just could be that they bind also, and it somehow stops the expression somewhere in that pathway. So it doesn't have to be that it stops it at the reception end of the transduction signaling pathway. It could be, after that as well. Any case, the last thing it says, this will lead to less mating and a decreased insect population that will consume less crops. Bottom line, if the males need a pheromone to find the females and there's just less of the ability of the males to do that, there's going to be less offspring and therefore less moths to eat the crops. And let's continue on to the question number 3, the first of the 4-point questions. Number 3 is the bufflegrass question. This is the ecology question of the 2025 FRQ. Let's get right to 3A. It says describe. Okay, we should love describe action verbs. Tell me something about the effect that removing a keystone species will have on an ecosystem. This is a straight curriculum knowledge question. Got really nothing to do with the blurb here other than the bufflegrass is an invasive species and there is a keystone species part of the place where the invading bufflegrass is occurring. But this is just a general question about keystone species, and we should know that keystone species are, just as the word applies, is key to the survival of an ecosystem. This one species provides many benefits to other organisms and may provide their roles, their ecological niches, okay? Maybe the excrement that they give off is something that other organisms badly need to survive. It could be the shade they provide. It could be the organisms they eat or the environment they provide by their own chemicals coming off. There's so many different things, but keystone species are species that keep the ecosystem stable and energy flowing in many directions from the autotrophs, the plants, through the consumers, the animals, in a stable way. And losing that keystone species is going to collapse an ecosystem. That's just straight knowledge question from the curriculum. And by the way, if you haven't looked at the curriculum, if you're studying for the AP Bio exam, good look at the curriculum given by the College Board. Look under Essential Knowledge of the AP Bio description of the course, and that tells you the things you have to know. So let's get back to the written part of my answer. So the written part here, 3A, for the describe action verb, removing a keystone species will collapse an ecosystem. Word for word what I just said. And again, it is word for word based on how they present that in the curriculum. But you could have said this many different ways. You could describe this species, a keystone species, a little bit differently. You could have described it's a species that many other species in an ecosystem depend on, possibly for energy or for their ecological roles. are defined by this one species, and you could also have to say part of that would have to be that losing that important role giver, so to speak, in the ecosystem is going to result in the loss of many or multiple species in that ecosystem and lead to it being more unstable. I have to come out and say collapse, but you can say that it's going to lower the stability. It's going to possibly kill off some species in that ecosystem. All right, so on to 3B. 3B of the bufflegrass question says, identify a control group the scientists should include in their experiments. So we like identify. Just tell me, you don't tell me why, just tell me, identify it. A control book, tell me a control group that would be appropriate for this experiment. So let's go to the experiment they're talking about, and they said in the second paragraph they conducted an experiment to determine, I'm right here, whether they could control the abundance of the bufflegrass population. Scientists identified several native grass species that, when grown with bufflegrass, might reduce the abundance. Okay, they also grew bufflegrass in the presence of several different native species in greenhouses in both non-drought or drought conditions. All right, so they had two test groups right now. Okay, they probably had more, but we don't know. But we do know we have bufflegrass mixed with at least another native species. Remember, bufflegrass, easy for me to say here. All right, bufflegrass. The bufflegrass was an invasive species, so it's not native. So we wanted to see how the bufflegrass would grow in the presence, and maybe there are some native species that would reduce it. So they wanted to check that out. And so if I'm going to grow bufflegrass with these other native species, I want to see how well it grows in comparison to what? See, I've got nothing to compare it to unless I have some control. So how much is the bufflegrass growing with native species compared to, let's say, if there was no native species, right? That's the easiest way to do things. So notice there's two conditions, though, or two major test groups. There's one in drought conditions, right? There's one in, we have drought conditions, and one in watered or non-drought conditions. So in my opinion, the best control, which would provide something to compare our test groups to, are they growing more or less with the non-native species, or that with the native species compared to the bufflegrass by itself. So we need to compare how the bufflegrass is growing with this other grass, and to know if it's being impacted negatively or positively, we have to compare it to, hey, bufflegrass growing on its own in both non-drought and in both watered conditions. In my opinion, you would need to say that you have to have a bufflegrass of some control type. I can't think of another here, but there probably is. But just growing pure bufflegrass by itself in both non-drought and drought conditions, so that you can compare how much more or less bufflegrass grows around the other native grasses. So that's essentially very important. Let me show you what I wrote to identify what I thought would be a good control. So I stated an appropriate control would include a test group of only bufflegrass in non-drought and drought conditions. And again, the reason for a control, number one, would be for a comparison. The second reason for a control is to eliminate other variables. When you mix grasses with bufflegrass and you don't have an appropriate control like bufflegrass growing by itself, there may be other factors that are affecting that growth of the bufflegrass. So we can eliminate that, so to speak, by comparison, okay, with just the bufflegrass. All right, so two reasons why there, but I think that's the most clearest and the easiest way to go about your business in terms of getting an appropriate control. On to 3C. 3C says state. There's our action word, state. Just state it, write it. The null hypothesis of the experiment in which buffalo grass is grown in the presence of native grass species. So the reasoning for this null hypothesis is to test it with statistics like a chi-squared. Okay, well, what we want to do here, what is happening in the experiment is we're growing buffalo grass with other native species to see if those native species will impact the growth of buffalo grass, since it is an invasive species. Will it limit its growth, or will buffalo grass be exponentially grow in those conditions? The previous question B was asking you to write a control. Here we want to write a null hypothesis. And again, to understand to write the null hypothesis is to understand the reasoning why we write null hypotheses. It's very difficult to try to measure what should be expected from an outcome of an experiment. We don't know what's going to happen. We can possibly predict some outcomes, but an experiment is not just getting results that we expect. Experiments are trying to gather data to understand what's happening. So what we do statistically to try to come up with significance, some quantitative ways, is we want to measure in a way that we're measuring from the outcome of no change. We can easily say, hey, there's going to be no difference, and then we can measure from that vantage point. So the statistic that you learned in this class, AP Biology curriculum, being the chi-squared analysis, we're going to measure how far away a expected and an observed are. If they're close together, the observed is coming out to the expected, we can say that, hey, there really is no difference. If there's a great difference, well, something other than chance is driving that. To measure that quantitatively, what we need to do is set a line in the sand for no change. We can't put a line in the sand for some change that we're not sure what's going to happen, right? You can't say, well, it's going to change in a way that's going to be this, and let's see how close we are. Well, we don't know what the change is, but we can know what no change is. That's the key here. So we're going to state the hypothesis that there's not going to be any difference, okay, in the outcome of our experiment with different grasses grown with buffalo grass. It's not going to have any effect whatsoever. And if there is a difference, we can measure how far it is from that baseline. That's important. So when I state the null hypothesis, essentially I'm saying that there isn't going to be no difference between the expected value, okay, which is in our case to the observed. We don't know what the observed can be, but our expected value is going to be, hey, some outcome that we're testing against. So in this case, since there isn't any real outcomes we're trying to test against, we're just going to say that the line in the sand is there's going to be no difference. That's the key here. No difference between those two. Now, they're not going to accept if you say there is no difference between the accepted and the observed. Okay, that will not be accepted. I know that for a fact from reading previous chief reader reports here. So what we can state is there's going to be no significant difference between the buffalo grass grown with different native species and with buffalo grass growing alone. And that would be one way of stating there will be no significant difference between the observed and the expected. That's important. You can also come out and be a little more, how can I say it, honors, or a little bit better in stating that, hey, there's no effect on native grasses on the buffalo grass growth or the abundance of the buffalo grass. Okay, so in any case, a couple different ways to state it, but you cannot expect to get the point by saying there is no difference between observed and expected. That's just not enough information. Put in the variables. There will be no difference. Just state it. I'm explaining the why part. You just state it. There will be no difference in the abundance of buffalo grass between it grown with native species and it by itself. Boom, done. Okay, now let me show you what I wrote. So state is my action verb, and I said the abundance of buffalo grass is not affected by growing with other grass species in either non-drought or drought conditions. Simple as that. And I didn't mention that when I first talked about that, but that's as simple as it gets. And again, if you want to come out and you want to be more, how can I say, mechanical, because you're not really used to writing these, write it in a way where there's no difference between what's expected and observed, but put the variables in. There will be no difference between the amount or the abundance of buffalo grass grown with other grass species in non-drought or drought conditions. All right, so it's as simple as that. Last question of the buffalo grass blurb here. So 3D, scientists have found that the population growth rates of native species are much slower than the population growth of buffalo grass following a wildfire. Now, if you remember back in the reading, if you looked, they said that buffalo grass is drought tolerant, which is a condition before a fire. So when you have a very dry area, buffalo grass is still growing and can survive wildfires. So both of those conditions suggest that it's going to live longer and during dry seasons and get past a potential fire. Also, it says that dry buffalo grass acts as a fuel for wildfire, causing the fires to be more severe. So buffalo grass can survive in drought tolerant conditions that provide and also provides the conditions for wildfires to occur and to make them even more severe and hurt more organisms. So with that information, look at the next question, or the next part of the D question. The scientists claim that the wildfires will therefore increase the abundance of buffalo grass plants in the ecosystem based on the information given. So when I look at this, I state that number one, that buffalo grass survives wildfires. That's really important. And it also fuels the wildfires. So it creates the conditions for wildfires and also creates the conditions for more severe wildfires. And therefore, they can, of course, kill off any of the native species that are competing against to take more of an active growth in its area. All right, but also something it says here that catches my eye is that the keystone species, this cactus they talked about, is a keystone species, which basically helps provide important ecological roles, or at least keeps the ecosystem together. It's the glue, so to speak, that keeps the ecosystem strong and stable, is susceptible, okay, to wildfires, especially the young ones. So they say the young ones, okay, cannot survive the wildfires, where the older ones will. Well, the older ones are eventually going to die off. So if you're going to have multiple wildfires because of the dry buffalo grass drying and of course providing the fuel and making the wildfires severe, you're going to kill off all of the young keystone species in this ecosystem. And just like question A talked about, the answer there was that you're going to collapse the ecosystem. Well, geez, you're going to lower, okay, the ability of the native grasses that are part of this ecosystem to grow on top of that. So to me, the answer, okay, probably could be accepted in talking about that buffalo grass can exist in drought conditions and is the cause of fires and survives the wildfires. To me, that's something that's important, but we don't know about the native grasses. But I do think talking about that alone probably is enough to get the answer right. But I think the most sure way to answer this question is to talk about the keystone species that's being eliminated in these wildfires. And we have to understand that the young cacti, the cactus here, that's the keystone species, if it is going to die out, well, so will many of the species it supports or provides ecological roles for, indirectly or directly. And so the native grasses would be part of that ecosystem. So for me, the best answer would be talk about that the buffalo grass helps produce wildfires that are killing off the ecosystem, or killing off the keystone species that is supporting the native grasses. So let's go see what my answer is. Remember, they did say that the native grasses do grow slower than the buffalo grass. So I said the higher rate of higher rate of growth of bufflegrass will result in increased dried biomass of fuel that will increase the potential of a wildfire. The wildfires will decrease the population of a keystone species, that's the cactus, leading to an unstable ecosystem that results in less native species to compete for resources with the bufflegrass. So for me, the best answer is to talk about that these wildfires impact the keystone species, which will negatively impact all the other species and collapse that ecosystem that, of course, is being supported, that the native grasses that we're comparing to the bufflegrass are not being supported now. They don't have the keystone species to keep that ecosystem together, and those native grasses are going to die out, which is going to give a better or a greater ability for bufflegrass to even gain a bigger stronghold. So again, for me, the best answer is that incorporation to the keystone species that's being negatively affected by these more severe and more frequent wildfires that would probably occur because of the bufflegrass. All right, on to 4. Number 4A, describe. The action verb we like, describe. Tell me something about the genetic evidence that evolution is occurring in a population. Keyword here is that we're dealing with a population. We're talking about evolution, which occurs between generations, and we're talking about genetic evidence. All right, so for me, looking at this question here, anytime I think about evolution, I'm thinking about the fact that the frequency of alleles is changing. That's the bottom line for evolution. Now they're speaking to genetic evidence. So when I hear of genetic evidence, I think, well, the best one is the DNA. And so if we look at the nucleotide code of the individuals in a population, is that of a certain gene that represents an allele, then we would say that, very simply, that is there a change in the frequency of the genetic code that supports a certain allele. What this is not, or what I do not believe is accepted, is that you're going to say, or if you were to say, well, there's going to be mutations. Mutations do occur. They're very slow. Careful with any type of mutation as your answer. They don't occur frequently. And evolution, yes, utilizes mutations that are already in the environment, and they're there to give the population some diversity. What's the diversity? Well, the difference in the amino acid sequence that results from a nucleotide sequence from a gene being different from each other creates new alleles, or creates different alleles. And that could be the tall allele or the short allele. If we're talking about gophers, that's my greatest example. So when I think about evolution, I always think about the frequency of those alleles or these different types of genes increasing or decreasing and changing. I don't think in terms of all of a sudden there's a longer neck. All of a sudden we have a flipper. Okay, that's what people tend to jump to. So the bottom line of evolution is the change in the percentages of the individuals that have certain types of the same gene. And since they say genetic evidence, we probably should speak to the fact that we're talking about there's a slight nucleotide difference in these certain genes. But I do believe that saying the frequency, okay, of the alleles would be enough. We'll see how this goes. So let's go to my answer. So I wrote in 4A the change in frequency of any allele in the preceding generation of a population. To me, that's the genetic evidence. You have a greater percentage or frequency of a certain allele, a certain what? A certain type of gene on the same type of chromosome that's occurring. And that is essentially the bare bones of evolution. You don't have to have a brand new allele, okay, a brand new mutation, which I know a lot of people would fall for to have evolution. You're just going to just change the amount of certain alleles. There's more individuals that are taller in the gopher population. That's evolution. Yes, there was a mutation down the line somewhere because you don't have taller gophers unless you have the genetic code to create that expressed gene or allele. So that has to happen first, and of course there has to be some selective pressure that supports a change in frequency, where the taller gophers now are going to be have greater fitness and greater ability to reproduce and live better than the shorter ones. So the genetic evidence is the amount of the alleles, which is really measuring the amount of a certain type of genetic code. So it is a little bit I wouldn't say misleading, but you could fall for, oh, the genetic evidence got to be mutation. So that is, in my opinion, the best answer there. On to 4B. 4B, explain. That is our action word here. We're going to explain how the isolation of a marine species by the formation of a land berry can lead to divergent evolution. Okay, well, first of all, we've got to know what divergent evolution is. Divergent evolution is when new species occur from a common ancestor. So if we're talking about a phylogenetic tree of some sort, there's a common species and there's species, let's say B and C, and A used to be the old evolutionary related species that both B and C diverged from. Okay, so understanding that, we're talking about how and explaining how this isolation can lead to this new species being built. We have to understand that creating new species means that we're creating a reproductive barrier. And how does that occur? Well, it's going to occur because there's going to be some evolution. And that evolution occurs because of the geographic isolation. Having these species now in a new region where there's new selective pressures, it's going to lead to the different alleles being selected, and possibly, if there's a mutation over time that develops, new alleles that develop. And that's going to essentially create a scenario where there's reproductive isolation that creates a species. That's important that we understand this divergent. So let's take a look at my answer. So my written words here say divergent evolution occurs in geographic isolation due to different selective pressures that each different geographic region presents. Natural selection results in divergent evolution between species in different regions as a result of these different selective pressures. In time, changes in allele frequency or new alleles from mutations that develop bring about reproductive isolation. To me, you really need to speak to divergent evolution creating an opportunity for this reproductive isolation. I understand that the species is going to be isolated geographically, but I would think you would need to explain the fact that over time, in different areas with different pressures, the evolution is going to occur that's going to create this isolation. So that's, to me, a very important part of this explanation. I don't think it's good enough to say that the geographic isolation, boy, that makes new species and that's it. No, the isolation in the different regions create new environmental pressures that are needed, and that over time, those changes are going to result in changes in possible and different ways that they reproduce, possibly different time, that'd be temporal. It could be different ways in which they attract mates, okay, because of the, different ecosystems they are now involved with, or the different environmental pressures. A whole list of things that we can talk about in terms of pre and post zygotic isolations that can occur. But the bottom line is there has to be some natural selection away from each other. That's what divergence means, okay? From each other, from a common ancestor, to make two new species. Therefore, to me, it's very important that I hear something about reproductive isolation in the answer here. All right. Any case, on to 4C. 4C, predict the effect the formation of the isthmus had on resource availability for South American species. So now we're going to predict. Okay, so that's our action word. We're going to try to come up with something that we think is going to happen based upon some information given to us. And since C and D are underneath this blurb, it's how they're doing things now. They're kind of letting you know where it's coming from. So we're going to have to go through this little piece of information here. So let's go read this and see what parts are probably very relevant here for this question. The formation of the Isthmus of Panama connected two continents, North America and South America. North America land animal species migrated to the South America after the formation of the isthmus and occurred similar niches as South American species. That to me is important. So the landmass connected the two continents occurred. That's the isthmus, I can't say it, all right? And that allowed the animals from the North America to go down to the South America, where there's going to be a heck of a lot more resources, right? South America is warmer, there's more plants growing, the ecosystems can support a whole variety of other things going on there. So there's a lot more energy that can flow up through the autotrophic levels, or the trophic levels from the autotrophs in a warmer species. Now, so there's a lot more resources down there for sure. So it makes sense that they would travel down there. Now, it says here, and occupied similar niches as South America. This is very, very vital to the question. This to me is the most important piece of information. So we know the animals traveled south and they occupied similar niches as South America's species. Now we should know the niche is a ecological role. It's a part of the ecosystem where they move the energy upward. Their job, what do they actually eat and what do they provide for other ecosystems? How are they connected in the energy level, the food web? What job of the ecosystem do they have? But they're obviously similar animals are moving down and they're going to look for the same resources, but they're saying they're going to occupy similar but not same. So when I think about this question, I think of resource partitioning. And whether or not you know that word or not, it is part of your curriculum if you look at the essential knowledge part of your curriculum. So I think of the idea that we're going to have these animals come down, and we know that the resources are now going to be somewhat limited for the other South American species, because now there's going to be competition. But if there's going to be similar niches but not the same, then it sounds to me that these resources are going to be partitioned in a way to limit this competition so that all species can survive. And so resource partitioning would mean that these land animals, although eating similar or using parts of the similar ecosystem, would now, to limit the competition and therefore reduce numbers of both species, maybe the different individuals now will use different parts of that similar niche. Maybe they feed off trees, and maybe one can now feed off of the higher part of the tree and one lower part of the tree, whatever it might be. So to me, the effect of the formation of resource availability would mean that there possibly could be decreased, but to me, the resources are going to be partitioned or divided in a way to limit the competition so that the species can both survive. If they were going to both share exactly the same niches, then what would happen is then there'd be direct competition, and some species will not survive. So it's important that we know that they did get down there, they did survive, and they, to me, this word occupied similar niches. So to me, that means that they somehow kind of broke up or divvied out different parts of the similar niches so they would limit the amount of competition so they both could survive. And remember, South America, you're getting close to the equator. There's just a lot more energy in these food webs to divvy out. We're not going north where there's going to be less and less. So that's why, of course, there's more organisms that live near the equator than you say to the north or the south, as the energy level from the sun decreases and there's less at the ecosystem. So there's going to be plenty to share from as long as we're not in direct contact. So let's see what I said here. So my statement or my written part of this answer in the predict action, I'm going to predict that the formation of the isthmus resulted in resource partitioning of the South American ecosystems as land animals from North America migrated south. I come out and just said resource partitioning because I believe that's what we're looking for here. And again, if you don't come out and say that word, you just said, listen, the resources will be divvied up in a way that reduces the competition so that both can survive, to me would be more than enough. I don't think by you saying the resources will be lowered is enough, okay, based upon that statement that we read, utilizing this statement here that we read in the question that said occupied similar niches. That to me is the key to knowing we need something like the resource partitioning going on, where we're divvying up the resources so we limit the competition. Okay, on to 4D. So 4D is asking you to justify your reasoning or your prediction of how resources would be used if the North American animals traveled to South America. All right, and since again, the statement to me very important is that the North American animals are going to share similar niches, or in this case, roles in the ecosystem due to similar energy they're going to obtain or eat, then it seems to me that key to this question is understanding that you need to talk about competition somewhere. Hey, they're going to compete for similar resources if they're in similar niches, but if they're going to survive, in my opinion, we're talking about resource partitioning in this question, divvying up the energy in different parts of the same niche so that different species can coexist without directly competing for those resources. So to justify the fact, we need to limit, okay, the competition so that both can survive. The idea that they know that North American animals migrated south sounds to me like they survived very well. And again, by saying that they were in similar niches, then they both were surviving. And the only way that could happen is if we limited competition. And the only way to limit competition for the same resources is they just found different regions or different areas of the same niche, or just slightly different parts of the same niche to limit that competition. So I said animals that normally compete for the same food or resources now have to divide the limited resources. Now, they may not be so limited. I think about this limited, maybe that's not necessary, but divide the limited resources by obtaining, I guess they are limited, there's only so much, even though you are in South America near the equator. Divide the limited resources by taming these resources in different regions of the same niche. I gave an example. They love to talk about the lizards. You used to see this question pop up in practice exams. For instance, lizards from North America that migrate to South America may eat the same insects in a certain tree and limit competition by living in different heights of the tree. Okay, so that's where I think this question was going. All right, on to 5. Number 5. A, describe. Describes our action word here. So describe a characteristic. Tell me something about the characteristic of an enzyme's active site. Tell me something. Describe it. Is it blue? Is it red? Tell me something. Describe. You should love to describe action word here. Okay, so tell me something about the characteristic of a enzyme active site that allows it to catalyze a specific chemical reaction. That's important, and we should already know that enzymes are very specific. So what about the enzyme's active site? Well, they have some pictures here, and they're kind of giving it away, in my opinion. If you look at enzyme 1, what's so special about this active site that I'm kind of coloring in red here that allows it to work with amino acid A? Well, it's got a complementary shape. There's some form and function. You think of proteins, you think of enzymes, form and function come into you. The form is the three-dimensional shape. The active site has to interact three-dimensionally somehow, has to be three-dimensionally comparable or comparable or complementary for it to interact. And that's important because enzymes or proteins in general are all about their three-dimensionality. How do they fold? What shape do they make? That shape, okay, is what drives the DNA code into action. I always say proteins are the soldiers of DNA. They get it done, but they get it done by their three-dimensionality. So a characteristic, they just want to describe a characteristic. So I would say very simply, if I haven't already said it enough, it's three-dimensionality, its form, its molecular shape has to be comparable to the substrate by which it works with or connects with or binds to temporarily to cause a chemical reaction to occur. So it has to have a three-dimensional shape. And you've seen this even when it's not enzymes. You talked about in different parts of your course about the transduction signaling pathway where the receptor has to, be complementary with the signaling molecule and make a ligand. So same idea here. So for me, it's very simple. It's a shape, and you may have learned about the lock and key method of an enzyme where it has a shape that can fit into a substrate to bind with it temporarily as it catalyzes the chemical reaction. Also, some small things you can also talk about is that besides having a complementary shape, you should know that this active site has a microenvironment that enhances whatever chemical reactions need to occur on those sites that are reactive. See, enzymes not only just bind to substrates, they cause chemical reactions because they set up a condition so that the reaction can occur, either by putting substrates close together in their active site or creating the environment that induces the reaction in a certain specific place on the substrate. So let's take a look at what I said in my written answer. So I said in the describe action verb here for 5a, the active site provides a three-dimensional form that's complementary to the shape of the substrate so that it can attract and bind to it. I also added for good measure, and of course they just describe one, they said to describe a characteristic, but I'm going to add another one here. It also provides a microenvironment that supports the chemical substrates and chemical reactions that occur. Okay, so that would be more than enough. Form and function is what this is really about, and that diagram really, I think, should have given it away for you. If not, hey, you'll learn from that. Okay, on to 5b. So 5b states, based on figure 1, the figure we're seeing right above the questions here, explain, that's our action verb. We're going to have to try to explain, try to come up with something that explains how the binding of amino acid A to enzyme 1, which is what's happening right here, is regulated. That's another important word, because regulated can mean increase or decrease by amino acid B. So if you look at enzyme 1 acting on amino acid A, if you look downstream, we eventually make amino acid B from those steps, of those intermediates along the way, including amino acid A adding as, starting as the initial substrate. So something downstream, like amino acid A, they're showing in the diagram, all right, will bind, look at that, will bind to amino enzyme 1, and you can look very carefully that it's binding not on the active site, although we don't see the cut out active site, but it's binding on an allosteric site. So this is going to be a non-competitive inhibitor, and we can see this diagram right here. It's showing that it basically is a diagram telling you that it stops that function. So something downstream, which is going to prevent the continuation of this metabolic pathway, which is a series of steps from occurring, it's going to stop this, is negative feedback. Okay, is negative feedback, and that's something you should recognize. It's on every test, and it's an important part of how we regulate things. If we're making too much amino acid B, it would be what? Disadvantageous of the organism to keep making it. So one of the ways that we do this in many metabolic pathways is have that product downstream act as an inhibitor, in this case a non-competitive inhibitor, and bind to the enzyme and change its active site. In fact, I said that it didn't show the active site because when this amino acid B bound to it, it changed its shape allosterically in such a way that the R groups attracted each other differently to change the active site. There is no active site here. All right, so the no active site is what that allosteric change caused, and so therefore amino acid A can no longer be used, and therefore this entire process stops. All right, so again, something downstream affects something upstream in a way that shuts it down is negative feedback. So many ways to discuss this, and the key to understanding is they're regulated, and looking at this diagram, I think it kind of spells it out. So, and they didn't say you had to say an allosteric change. They didn't say specifics in terms of this, but the binding is regulated by amino acid B. They want you to explain how that occurs. And let's get into that with my answer. So for 5B, my written answer says the binding of amino acid A to enzyme 1 is regulated by negative feedback through the allosteric change that enzyme 1 undergoes when amino acid B acts as a non-competitive inhibitor. Okay, but again, if you didn't use non-competitive, you could have just kind of, in my opinion, you could have discussed the fact that the amino acid B would bind to the enzyme 1 and then change its form in a way that its active site is no longer available, and therefore shut the process down. I'm saying it in less words by using negative feedback, non-competitive inhibitor, allosteric change. So you could describe that certainly different ways with more words. All right, so essentially that's the answer there. That's definitely the answer to that question. On to 5C. 5C, using the information in figure 1, that's the picture above, identify. Ooh, we like the identify. Love the identify action verb. Just tell me, find it, identify it. There's no discussion, there's no telling me why, just go find it. So they want you to find in figure 1, identify the product of the reaction catalyzed by enzyme 2. So we find enzyme 2, and the product, what's being produced, my friends, is intermediate Y. And they even gave you the choices. I found it to be very interesting. So using information in figure 1, identify the product of the reaction catalyzed by enzyme 2, intermediate X, intermediate Y, or amino acid B. So if you had no idea, you could have made a 1 in 3 guess here. So clearly you shouldn't really have to. Hopefully you can see a product is what's produced after. This would be a reactant if that's what got you down. Reactant is what you start with. Intermediate X is the reactant. The enzyme takes this substrate, causes a chemical reaction, and then we produce after. That's the arrow after. That's the intermediate. Okay, so intermediate Y is the answer. And there it is, written out, not a big deal. Just figure out what was produced, what was coming after that enzyme. On to 5D. 5D states, based on figure 1, the picture above, explain. Okay, we need to somehow discuss in a way that's going to support this question, how a change in pH could affect enzyme 3 in such a way that amino acid B cannot be produced. So we're talking about enzyme 3. And if we're talking about how a change in pH could affect enzyme 3, well, we should know the factors that affect enzyme ability. Of course, we should know temperature, and too high of a temperature can break the R groups and actually break bonds, and we call that denaturing. Well, this idea of denaturing, okay, or denature, occurs at pH 2. All right, so if the pH is too low, of course that's an acidic range, denaturing can occur, and if the pH is too high in a more basic range, denaturing can also occur. We should recognize the fact that if we were to plot a pH change and the activity of an enzyme, there's going to be an optimal pH range, whatever that might be. But if you get too low in pH or too high, okay, the pH eventually lowers and stops working because a caustic or acidic, okay, types of solutions will actually break bonds beyond breaking down the attractions of the R groups. So that's really important here. So this question is really about, do you understand that pH is a factor that affects enzyme activity? So the answer to me, you could have a pH that's outside of that optimal range, meaning it could be, like I drew, too high, too basic, or too acidic, and being outside that optimal range would eventually lead to a denaturing, which will change the shape of the enzyme, including its most important active site, so much that intermediate Y will not be able to interact and bind with it temporarily, and therefore the reaction does not go to produce B. Let's see what I wrote for 5D. So I said in 5D in explaining how the pH could affect enzymes 3's activity in terms of producing the amino acid, the product, the final product. I said a change in the pH could affect enzyme 3 if the pH becomes too acidic or too basic. I'm describing here that there is an optimal range outside that optimal range. I could have gone and said that, but I went and talked about the pH going down and pH going up to talk about this important, graph of optimal pH activity. Different proteins and enzymes will have different ranges they work best under. Under non-optimal, I guess I did talk about that. Under non-optimal pH, an enzyme can denature to a point that the active site changes its form and thus its function. Here we go with form and function. So much that it cannot provide an active site that will bind or interact with intermediate Y. And if it doesn't bind or interact, then the reaction isn't going to go. All right, so again, that's really an important understanding, the fact that we're going to change the shape to a point where the active site no longer can be complementary with the substrate, so therefore the catalyst doesn't work anymore. All right, so on to number 6. 6A, the final FRQ question, final 4-point question here, states, based on figure 1A, identify, that's our action verb here, identify, I like that one, just tell me the fly genotype in which the average percent metaphase cells with the ALD associated filaments is close to 12%. So we have the genotypes listed here on the x-axis, and we're measuring the percent of the metaphase cells of these flies that have the filaments that are necessary for successful reproduction. Okay, so we look and we see, very simple, I want something around 12%. So we look at the halfway mark here, and you could probably eyeball and see that this value here for this genotype is below that halfway. And if you look at the genotype ALD1 DEL, you can see that this genotype, okay, basically is right around that 12%. If you want to consider the error bars or not, interesting enough, they did not even mention what the error bars were, so you really can't use them here, but the value of this bar graph, okay, in my opinion, is very close to the 12, where nothing else is. Even with the error bars, in my opinion, okay, this all 23 is not a solution that represents the halfway. And again, I know that is tough to look at the way that I drew that, but if I do that more carefully, and again, they want you to pay particular attention, the halfway mark I'm again estimating But you could, now again, we just on the computer now. So we're a little bit past the halfway mark. And if I just kind of keep going here, you can see that we're under the halfway mark. I know it's not a great straight line that I'm drawing there, but I think you can eyeball that and see that the ALD23 genotype is below the 12% or below the halfway. So the only good choice you had there, in my opinion, was this one. And they just wanted to identify the what? The genotype. And if you didn't know what genotype was, they're telling you the all genotype right there. That's what all these are here. Okay, now the phenotype would be the expressed amount of the gene, and that's what is actually being measured here on the y-axis. How much is expressed, right? How much of the cells are making that filament with the ALD protein that they need to be successful in. So let's kind of zoom back to the question. So we're just looking for which one is closest to 12% right off the graph. I think that's something we can definitely, and I call that a low-hanging piece of fruit. Okay, that is definitely something that we can and should have gotten that answer from right off the graph. So let's see what I said here in my written response. So I said for the identify action verb for 6A, I said the ALD1 DEL. That's the genotype. That represents the genotype that has the average percent metaphase cells closest to 12%. You really don't need the rest of that, but just do that for understanding what question you're reading. Okay, so on to 6B. So 6B says, based on figure 1B, okay, which is a protein assay there. You're just seeing how much protein's being made. Okay, describe, that's our action verb, the difference in the ALD protein production between gamete-forming metaphase cells with the genotype ALD3 and ALD23 and the ALD23 and the DEL. So using B, we can see that there is some small amount of protein being made with the ALD3 and ALD23, but no measurable amount for the ALD23 DEL. Now, if we're not sure what B means, okay, they did come out and say that a thicker band indicates a greater amount of protein. Well, we've got a band that's showing some protein, and we have no band here, which means there's no protein. So that comparison means we're producing some amount for the ALD3, ALD23, but no protein, no visible, measurable amount of protein, ALD23 proteins being made there. So let's go on to describing that in my written part of the test. So I'm describing that difference. I'm not explaining, and I'm just going to say that the ALD3, ALD23 does produce all proteins, ALD proteins. That's the protein, that's the helpful protein in helping in metaphase and helps pull apart them in anaphase to the right chromosomes. While ALD23 DEL does not produce a measurable amount or does not produce any. You could say ALD23 produces some and ALD23 DEL does not. Just reading right from that second part of that graphic. Okay, so it's as straightforward as that. Another piece, in my opinion, low-hanging fruit to get that point. Okay, so if you got confused somewhere, it's just as easy as that. On to 6C. So 6C states scientists hypothesize that gamete-forming metaphase cells can produce a normal amount of ALD-associated filaments even when they produce, and this is a key part here, even when they produce about half as much ALD protein as the wild-type cells produce. Use the data in figures 1A and 1B, they're telling you, to support the scientists' hypothesis. So we're going to have to use the data to support the hypothesis. What is that hypothesis again? Rereading and rephrasing these questions really are helpful. Scientists hypothesize that gamete-forming metaphase can produce a normal amount, even when they produce half as much protein. Okay, so understand the difference between A and B. A, we're looking about the percent of cells with the ALD filaments. And when we look at the wild type, that might have gotten some of you. Wild type means that we're going to not have any of the recessive alleles, WT and WT from the reading. are your dominant alleles, okay, or dominant chromosomes. This is a heterozygote that has the deleted chromosome gene, but still has one chromosome to still produce the same amount. If we're talking about plus or minus 2 SEM, we don't know what they are in this question. But so the idea is that there is statistically the same amount of metaphase cells with the associated filaments between the wild type, okay, and the one with that has, you know, has a recessive allele there, okay, in its zygous in the heterozygotes. Okay, any case, so when we compare the fact that there is twice as much protein being produced in the wild type than the heterozygote. So we have, again, this data supports the idea, let's get back to it, the idea that we can produce normal amount of cells. So we have this essentially the same amount of cells when you consider the error bars, whatever they might be. So we have the similar amount of metaphase cells with the correct ALD associated filaments, even though, okay, the heterozygote produces half as much proteins. So there it is. And that's understanding the differences between the data. This is about the percent of the cells that have that good protein, and this is the amount of the protein. So that statement is definitely supported, okay, or its hypothesis is supported based on understanding that data. Let's go see what I wrote. So I'm again thinking about what my action verb is, and I want to support that hypothesis that we can have a normal amount of cells in metaphase with the correct protein, the ALD protein, but still produce half as much of it. And again, that came from the two different graphs. Now, if I'm going to support, they're telling us also to use the data. So I'm going to get specific here, as you should have. So I said in figure A, the wild type, that you could just said the wild type, and the heterozygote contain no significant difference in metaphase cells with ALD filaments, while the heterozygote genotype produced half as much ALD protein based on figure B. So that directly supports exactly what the hypothesis stated. Figure B illustrates this difference by the ALD protein bands of WTDL, which is the heterozygote, being half the thickness. I went on to describe that thickness, although probably was not necessary that statement. So I got really specific about A and B and what was happening there. So I got specific what was happening in A and B. Again, last statement probably was not necessary. On to number 6D. 6D, for gamete-forming metaphase cells of the heterozygote, at least the WTDEL genotype, and the ALD1-DEL genotype flies, explain. So they're telling us which ones we're looking at, okay? And these are for the gamete-forming metaphase cells. So we're talking about the figure A, okay, that bar graph. Explain why the phenotypes observed in figure 1A differ, even though the amount of ALD protein produced in figure 1B does not. So let's take a look at WTDL and ALD1DL. WTDL and ALD1. So let's clear this up a little bit here. So when you think about this, we're going to compare the WTDL and the ALD1DL. And we can clearly see from figure B that we're approximately making the same amount of these ALD proteins that are used in the filaments and the centromeres that help pull these chromosomes apart to make these gametes, right? Gamete producing cells that will lead to new offspring, right? So we're clearly making the same amount with one good chromosome and then these two recessive here. But the question states, why They want you, again, don't forget what, if you keep looking, what are they talking about? They want you to explain. So it's not just good enough to tell me here. So we need to explain why you think that we have the same amount of protein being produced, yet when you look above, okay, very clearly, the amount of metaphase producing cells in each of these is so far different. Having the one wild type chromosome allows for a heck of a lot more cells having the ALD associated filaments when being homozygous recessive for this type of recessive chromosome or allele. There were so far fewer. There was significantly less of the metaphase cells. How can that be if we're producing the same amount of protein? Why is there so far fewer cells with the ALD1 and DEL genotype? Well, this, again, if you're doing practice tests in the previous test, okay, this definitely got me when I was reading it. But if you go back to the question, they gave you some important insight here. In the absence of functional, and this is important, ALD proteins, gamete-producing cells enter anaphase 1 before homologous chromosomes are correctly aligned. And what's that going to happen? As a result, gametes produced do not contain the correct number of chromosomes. My friends in chemistry or my friends in biology, I keep doing that because I teach both. We're talking about non-disjunction here, non-disjunction. So if you didn't pick up on that, okay, you need to look at the previous year's FRQs. It's definitely a parallel there. So what does that mean for us? Well, that means is we're making the same amount of what? Protein, even though there's fewer cells that have the filaments. Well, how can that be if there's far fewer cells with the filaments that we're producing essentially the same amount? Well, there's got to be cells that have extra chromosomes, right? Because of non-disjunction from this extra protein, or the protein that's equal here, is due to the non-disjunction that produces a greater number of chromosomes in these individuals. So if you have multiple duplicate chromosomes, which is occurring in the non-disjunction because you don't have the correct, or don't have a lot of the correct protein, then you have, again, duplicates of chromosomes that do have the ALD that make up for the cells that just don't. Okay, so the cells that are getting duplicates could possibly be producing more of the ALD filaments to make up for the low number of cells that don't have it. Okay, so that's the explanation there. So what did I say? Let's see what I said. So my written response for 60 using the explain action verb, I said the percent metaphase cells with ALD filaments differ significantly with the WT-DL and ALD1-DL in figure 1A, but demonstrates equal amounts of ALD protein produced in figure 1B because the ALD1-DL individuals produce a low percentage of cells with ALD protein, but demonstrate a similar amount of ALD proteins. This is due to the large number of surviving gametes that have extra chromosomes, I said N+1, due to non-disjunction. Okay, so that's the explanation. The similar amount of protein produced is due to the surviving cells that had multiple chromosomes with the ALD protein that overcame the low number of metaphase cells in the first diagram. I hope that helped, and good luck studying for this AP.